So this is a podcast on DNA replication. After completing the POGO, you should have learned a few things about the structure of DNA. So here on the right side of the screen, you see DNA. And here on the left side, you see something called a nucleotide. And if you guys look closely, this nucleotide, we could think of it as a Lego. And when you put Legos together, you create a structure. So if you look on your right, you see that same Lego here, 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 and here. So you should know that DNA has a chain of these nucleotides, and it has two strands, two chains. Each nucleotide has three parts. Each Lego has three parts. It has a phosphate group. It has a sugar. This sugar is called the oxyribose. It's a 5-carbon sugar. And right next to it, there's a nitrogen base. Okay. There are 4 different types of nitrogen bases in DNA. The first one is thymine. The second one is adenine. The third one is guanine. And the fourth one is cytosine. When you put them together, you generate strands, and these bases then become connected by something called a hydrogen bond. Then it twists like a ladder and it creates a double helix, which is DNA. So here's your DNA. Here again, in different colors, you see the different bases. But these bases are always paired up. These bases are always paired up in the same way. G always pairs with C, A always pairs with T. A shortcut to remember it is, I like to call it the G-cat, the gangster cat. So the G pairs with C and A pairs with T. Okay, so that's one way to remember it. These base pair rules are also called Chargaff's rules. So you might see it as base pair rules, or you might see it as Chargaff's rules. And again, just like I said before, the nitrogen bases, which are these guys right here, are held together by hydrogen bonds. So if you take chemistry, you will learn a little bit more about hydrogen bonds. They're the forces that hold the two strands together. So if we go back, these are hydrogen bonds right here, these little dashed lines, and they hold the DNA structures together. They hold these nucleotides here are basically held together by the hydrogen bonds. And here it is. Here's another picture. Here's your phosphate group. Here's your sugar. Here's your base. G-cat. A pairs with T. G pairs with C. And again, there's two strands. And these two strands of nucleotides are held together by hydrogen bonds. Okay? And so that's what you should have gotten from the structure of DNA. Now we're going into how DNA is duplicated. So how do you duplicate? How do you make more DNA? And this process is called DNA replication. So DNA replication is how you make more DNA. So if you look at the picture, if you get a little bit confused, everything is here. But I'm going to explain it using the picture. So again, we just learned that DNA has two strands. And during the DNA replication process, the way it works is that it starts to unwind. So the original DNA strand, which is in blue, is going to unzip. So here you see that it's unzipping, right? So it's separating. Then these bases, these nitrogen bases, become exposed. And what happens is that an enzyme called DNA polymerase comes in, can't see it here, but it comes in and it starts to add bases, new bases, and these new Legos, these new bases are in yellow, okay? And you see how the new bases are forming a new strand here? Okay, this is a new strand, and this is an old strand in baby blue. Okay, they're following Chargaff's rules. T pairs with A pairs with T. Okay, the same rules apply here. And towards the end, you're going to end up with two new DNAs, with one strand being new and one strand being old. Okay, and so this is how DNA replication works. And if you didn't catch that, it's here in writing. You just have to look at the picture and try to match it with what it's saying. And then on the next slide, once you make your two DNA strands, we noticed that one strand was new and one strand was old. Okay, we call this semi-conservative. Okay, semi-conservative, meaning that you're not making two new strands, you're only making one new strand for every old strand. Okay, so here it says here, when DNA polymerase is done creating complementary pairs, each old strand has a new partner. So at the end, we end up with two DNA molecules instead of one, but one of the strands is from the old strand, the original strand, the parent strand. We call this DNA replication semi-conservative because it keeps one of the old strands and it has a new strand. Okay, and here, it's just a little bit of practice. The following is a DNA strand that has been separated. So I basically drew this showing that it's separated, and it's asking for you to write the complementary bases. The complementary bases that DNA polymerase would add. So if this is one of the old strands, the new strand is going to be C is going to pair up with G, A is going to pair up with T, A is going to pair up with T, T is going to pair up with A, C with G, and then G with C. Okay. Now I want you guys to pause this podcast and try doing this one. Okay, for this one, find a complementary basis for this strand. Okay, now that you tried, here's the answer. G is going to pair with C. T is going to pair with A. T here is going to pair up with A. A is going to pair up with T. G is going to pair up with C. And C is going to pair up with G. Okay, so this right here, this and this, so let me highlight it. This and this is going to form one new DNA. Again, one old and one new. And then this one is your old one. And it's going to pair up with a new strand, and this one's going to be new. And again, this is one DNA and this is a second DNA. And once more, this process is semi-conservative, meaning that it keeps one of the old strands. Okay. So this is DNA replication. You're going to need this information for your comic strip.